Okay, could somebody confirm that you can hear me?
We can hear you.
Thank you. There we go. Zoom's showing an error message. What's going on here? I don't see anything here. Let's see, I'm recording, so let me share my screen. Don't know what that error message was. So, anyways, I had, well, before I begin, are there any questions? Not hearing anything, so let's move on. Let me go to the syllabus. Today is January 30th, and we should be working on chapter 4, prokaryote and eukaryotic cells. Any question about that? Let me state that I did do some grading. I can't show you the grade book now. I think lab 3 is graded for everyone for the first time. In lab 4, I have it graded for about half of you. I have postponed when I'll do the second grading of lab 4. It will not be this Saturday. It will be a week from this Saturday. And I think that's something like February 7th. I did get some grading done in the worksheets as well. With the worksheet, if you aren't sure of your answer, you should look it up because most of the time you'll only be answering one question. So you may as well get it correct. So if you're not certain, you should look up the answer in the lecture worksheet. Actually, you should look up the answers in the lab worksheet as well, but obviously it's going to take you more time. All right. Any questions about any of that? Let me state that I did do the extra credit study guide, and you guys need to take a look at what the I don't know, procedure or the rules are for the extra credit, meaning what you can work on and what you cannot. You cannot study this week's lab, meaning the current week's lab. I suppose you could study the current week lecture worksheet. I stated you shouldn't be studying that one either, but you're only answering one question, so it'd be very hard to actually study one question and try and get the answer there. But if you study the worksheet for the lecture, I'm not going to worry about it. Um, and then you should give the names of everyone in your study group, and then everybody should send in a uh, a um, a file stating what you studied, when you studied, for how long you studied, and everyone, the names of everyone in your group. You should also do some questioning. All right, any question about any of that? We were talking about the cell walls. Let me go back a little bit and gave the summary of the difference between a gram-positive cell and a gram-negative cell. Come on. Where did I put that now? I totally moved. Tried to blow it up, and then it wasn't on the page. There it is. So let me shrink that down now. Well, I can't see it, but there we go. We looked at the differences between the gram-positive bacteria and the gram-negative bacteria, and then talked about some differences between the cell walls of each. In the next slide, I need to talk about some cells that have atypical cell walls. First, let me talk about the domain archaea. It is a different domain of organisms than the bacteria, so it's not surprising that they have somewhat different cell walls. Instead of having peptidoglycan in their cell walls, they tend to have cell walls made up of pseudomurine. It's another disaccharide, and some of the archaea even lack cell walls, although I did mention that some of the bacteria lack cell walls as well. The archaea are really different in their cell walls and in their cell membranes. I'll talk about that in a future lesson. And it's just because they're a different domain. The bacteria and the eukaryotes have actually the same cell membrane. And it's thought that the eukaryotes just worked what was working well. And so they used the cell membrane of bacteria. You don't need to know that now. I'll talk about that later. But anyways, the point is the archaea have different cell walls. And there are some bacteria that have different or atypical cell walls. The Mycoplasma, that's a family of bacteria that have atypical cell walls, and they lack cell walls. They only have a cell membrane. They have sterols in their plasma membrane, and it's something similar to animal cells having cholesterol in their plasma membrane. So the sterol gives strength and support to the plasma membrane. Note that this sterol is not cholesterol. Cholesterol is only seen in animal cells. It is not seen in bacteria. It's another sterol, probably similar to cholesterol, but it's a different sterol. And then the other bacteria with an atypical cell wall is the Mycobacterium. I think we've already talked a little bit about this. It is a bacteria that has mycolic acid in their cell wall. And the mycolic acid is the main molecule in the cell wall of the Mycobacterium. All the other bacteria that have a cell wall, the main molecule in the cell wall is peptidoglycan. And there's a lot of peptidoglycan in the Mycobacterium, but there's a lot of mycolic acid, a little more in their cell walls. The mycolic acid give the Mycobacteria acid-fast staining properties, and we talked about that a little bit. Any question about anything I've discussed here? All right, didn't hear anything, so let's move on. You can damage the bacterial cell wall. You don't need to know the names here, but you can damage the cell wall with the enzyme lysozyme. It will digest the peptidoglycan or the disaccharide in the peptidoglycan. So I guess only the glycan part, removing the cell wall. And you can also damage the cell wall, at least the bacterial cell wall, with penicillin G. It's an antibiotic. You don't need to know the names of the wall-less gram-positive and the wall-less gram-negative cells, but if you do damage the cell wall to a bacteria, if the bacteria is in an environment that has higher water outside the cell than inside the cell, then that will eventually kill the cell, which is actually how penicillin G and the other members of the penicillin family work. They damage the cell wall. We'll talk a little bit about that, but they prevent the cross-link, the short peptides from going across, linking the NAG and NAM rows, and that creates a hole in the cell wall, and then the cell membrane can protrude out of that cell wall where the hole is, and then more water will move in, and the cell membrane will protrude further out of the hole in the cell wall, and eventually enough water will move into the cell that the cell membrane will expand too far, and then it'll burst, and that will kill the cell. That's actually how the penicillin family of antibiotics work, and that is most bacteria, when they infect a person, the bacteria is in a environment where there's higher water outside the cell than is inside the cell. And so water moves into the cell. And that's how penicillin can um, kill the bacteria. Any question about any of that? All right. I did mention that the gram-negative bacteria are more difficult to kill with penicillin G, so they're not as susceptible as the gram-positives to penicillin G. And it's because the gram-negatives have this outer membrane, which prevents the penicillin G from getting into the cell and damaging the cell wall. Any question about any of that? Some of the gram-negatives are not as susceptible to other members of the penicillin family or the family closely related to it, which I'm drawing a blank what that family is. Um, However, that doesn't mean that the gram-negative cells are not sensitive to antibiotics. They are sensitive to antibiotics that can penetrate the outer membrane, such as streptomycin. Streptomycin is a lipid-soluble antibiotic, and so it'll go right through the outer membrane of the gram-negative cell and then damage the gram-negative cell. And then you can have other antibiotics like ampicillin, which is actually chemically modified so that it can damage both gram-negative and gram-positive bacteria. And the ampicillin is one of the early antibiotics of the penicillin family that was discovered. It might be the third. If it's not the third antibiotic that was discovered in the penicillin family, it's close to the third. So it was an early member of the family to be discovered, and it can kill both gram-positive and gram-negative. Most of the later members of the penicillin family that were developed can kill both the gram-positive and the gram-negative bacteria. But penicillin G, the first member of the family, cannot. Any question about any of that? If not, let's change gears a little bit and talk about the plasma membrane. Remember, this lesson is talking about cell structures in prokaryote cells, and then later we'll talk about prokaryote structures in eukaryote cells if we have time. In the plasma membrane, the main molecule in both the bacteria and the eukaryotes is the phospholipid. And the phospholipids just get together and then form a bilayer. We get together like that, and then they form a bilayer. Let me blow this up. Like this, each one of those being a phospholipid, where the fatty acid portion is on the inside because the fatty acid likes being around other fatty acids or around other lipids. It does not like being around water, and there's a lot of water in the cytoplasm, and then a lot of water outside the cell. Think of this like a red blood cell. The blood is mostly water, and so the fatty acids won't want to be around the blood because it's mostly water. And what they do is they orient themselves so that the head of the phospholipid is facing outside in one layer, and then the head of the other layer is facing inside the cell. There are other molecules in the plasma membrane, including carbohydrates and proteins. With the proteins, there are three proteins you kind of need to know, and they're really defined mainly by what they are in relationship to the cell membrane. There are peripheral proteins. Let me blow this up. Let me see if I can go to the slideshow and blow this up. There we go. So a peripheral protein is a protein that is mostly on one side of the membrane or the other. It can have a portion in the membrane, but the majority of the protein is on one side of the membrane or the other. There's an integral protein, and that spans the membrane. And then transmembrane proteins, they also span the membrane. And this looks like a channel protein, one that makes a channel through the membrane to allow molecules to either enter the cell, come on, my mouse died, to either enter the cell or maybe molecules to leave the cell. Any question about any of that? All right, the plasma membrane is the gatekeeper to check what can come into the cell and what can leave the cell. The plasma membrane is said to follow the fluid mosaic model of a membrane. And the mosaic is that the cell membrane is made up of different molecules, phospholipids and proteins. So it's a mosaic of these different molecules. The main molecule being the phospholipid. And then this membrane is as fluid as olive oil. That's always amazing to me that I'm an animal, so I only have a cell membrane holding my cells together in my finger here. And what's holding them together is actually a fluid, which is just surprising to me because my hand doesn't feel very fluid. But the plasma membrane is a fluid, and it is as thick as olive oil. As you know, olive oil um, is not that thick, but it is thicker than, I don't know, canola oil or something like that, or even water. And as you were to put olive oil in the refrigerator, you'd see that it would start to solidify. But in the temperature of our hand, the cell membrane is a liquid, much like olive oil would be a liquid at the temperature of our body, 37 degrees Celsius. In fact, olive oil, if you don't know, is a fluid at room temperature as well. The proteins move about in the membrane to function. Some of you have taken A&P know that when a receptor protein binds to its ligand, the receptors tend to move together, and then they'll be taken into the cell altogether. So it's not just one that binds to the ligand and then that one receptor protein moves into the cell. They tend to move together in the cell membrane, and then they're taken in all together. Any question about any of that? Not all proteins in the cell membrane move. If the protein is anchored by either the extracellular matrix or the intracellular matrix, that protein will not move about in the cell membrane. But otherwise, the protein is free to move about in the membrane. Like transport protein, generally they're free to move about in the membrane. And so you could see it at the top of the cell, and then you could see it start moving down the cell. I suppose in theory it could move all the way down to the bottom of the cell. You could see it there later. Think of something like a cell in a bone cell in the bone. The bone has an extracellular matrix giving the bone its strength. The cell itself doesn't give the bone its strength. And you could have a protein in the cell membrane, which is anchored to the extracellular matrix. So I guess we'll make that here, make this attached to something here, extracellular matrix like part of the bone. That protein is then not going to be free to move about. It's going to be anchored in place. And then the same thing over here, if the protein is anchored by the intracellular matrix. That's a scaffolding of proteins running about the cell, making kind of the backbone of the cell. We'll talk a little bit about that later. But if the protein is anchored to the intracellular matrix, then that protein is also not going to move about in the cell membrane. Any question about any of that? All right, let's talk about the phospholipids. They also are free to move about the membrane. Why we call it the fluid mosaic model of the membrane, because the molecules are moving about as if the membrane is liquid, and at body temperature it is liquid, by the way. The phospholipids are free to rotate around, so just sort of spin in place, and they're free to move about laterally. Actually, the movement of the phospholipids are actually one way that water can enter a cell, and that is this phospholipid, this molecule right here, might move over here. That could make a gap right there. And let me blow that up even further. So right here we have a gap. Water can come in. It only goes about halfway because the water can't get through the fatty acid tail of the second bilayer of the cell membrane, but the water just stays in place and it occupies this spot so that the phospholipid cannot now occupy the spot. And then it just sits there waiting until this phospholipid moves over, and then the water can actually move into the cell. And that's one way that water can move into the cell. However, this movement of water into the cell is not very quick, not very efficient, and that is the water can move across the cell membrane, but not very fast. And most cells, especially or cells of the root, they have aquaporins, which are a transport protein that actually move water into the cell. I don't think the aquaporins are transport proteins. For one thing, like a root cell sending too much ATP for each molecule water you move in, because the plant usually needs lots of water. So lots of water molecules are moved into the plant. And the aquaporins are used by many cells to obtain the cell's water. Any question about any of that? All right. When we're talking about the cell membrane, it's important to realize that the cell membrane or the plasma membrane has selective permeability. The cell membrane is just another term for the plasma membrane. Selective permeability means that the membrane allows some things to pass through it and then enter the cell, but do not allow other molecules to pass through it. And that's why the cell membrane is considered the gatekeeper of the cell. Now, the plasma membrane has two, well, actually more than two, three ways of moving molecules into the cell or moving them out of the cell. Um, with the plasma membrane, it does not allow any large molecules to move into the cell unless the cell has a transport protein to transport the large molecule into the cell. For example, some cells can naturally, readily take up DNA. DNA is a very large molecule, so it cannot move through the lipid bilayer of the plasma membrane. But if the cell has a transport protein for DNA, it can then transport the DNA into the cell. Ions either do not move across the lipid bilayer, or if they do move, they move very slowly. And that's the molecule with a charge on it. That charge does not like moving through the lipid bilayer. Any question about any of that? Oop, I'm going to go further. Lipid molecules are free to move across the plasma membrane. So any lipid that is outside the cell can move through the cell wall and the cell membrane because the cell membrane is lipid soluble. So the lipid will move right through the cell membrane. Smaller molecules can move across the cell membrane, such as water and oxygen, by simple diffusion, where the molecule moves from areas of high concentration to areas of lower concentration. And like within the air, you always get simple diffusion going out so that eventually all of the molecules of the air that were a different smell or a different chemical composition, it'll be totally diffused so that the air would eventually have very little, if any, antibiotic or whatever we're moving into the cell. Any question about any of that? So lipid soluble molecules can move. Smaller molecules like water and oxygen can diffuse right through the cell membrane or the plasma membrane. Carbon dioxide can also do that, although carbon dioxide usually doesn't move into a bacteria cell or a eukaryote cell. The difference is if the organism is photosynthetic, it then can absorb CO2 and can do it either through aquaporins, which most plants have aquaporins, but a few do not, in which case they must get their water from um, a molecule moving through the cell membrane. Uh, actually, water is not. Oxygen and carbon dioxide are nonpolar molecules, and so they can move right through the cell membrane. Water, as I mentioned, does not easily move through water. It can move through water, but not very fast. And what happens is like I said, one molecule moves, and then a water molecule can occupy that spot, and it can't get through into the cell because the second layer of the bilayer will prevent it from getting through. But eventually, that molecule will, I don't know, give up the ghost so that it can move through. We're talking about water, and what happens is the waits for the lower phospholipid in the lower bilayer of the outer membrane. It waits, or the cell membrane, waits for that molecule to move out of the way, and then that molecule can move in and Those are different ways that the cell can obtain molecules, depending on what the molecule is. Oh, come on. I'm gonna have to end my show here because I can't get the mouse. Oop, didn't want to do that. There we go. When we're talking about the plasma membrane, it's important that there are some unique functions or properties of the plasma membrane. For one thing, the plasma membrane of bacteria contain the enzymes for ATP production. Eukaryotes make ATP in their mitochondria, but prokaryotes do not have a mitochondria. And so how they make their glucose is that they have the enzymes for making ATP in the plasma membrane. Let me blow this up. And if we were to look at the plasma membrane here, you'll note that it's uh, infolded. So the plasma membrane right there goes this way and then out. I'm going to blow it up much bigger than it is. And then pinches. And then it usually ends where it pinches. Let's go to the next one. Oh, I'm not in the slideshow anymore. That's why it's not working. So the enzymes for making ATP for bacteria are in the plasma membrane, and it's an infolding of the plasma membrane. The photosynthetic bacteria also have the pigments and the enzymes for making the end product of photosynthesis, usually sugar. Uh, those enzymes are also found in the plasma membrane. So for a bacteria, if it's photosynthetic, it will have the enzymes for the starting aerobic respiration inside the room where they usually measure somebody's height. You can damage the plasma membrane. If you do, then the contents of the cytoplasm will leak out of the cell, and that can kill the cell. You can damage the plasma membrane with alcohols. Alcohol will actually dissolve the lipid bilayer that we're talking about. The plasma membrane is made up of phospholipids, and alcohol will pull out the phospholipids, destroying the plasma membrane. You can also destroy the plasma membrane with quaternary ammonium compounds. These are compounds that, at least they used to be, they probably still are used frequently in hospitals to clean somebody's skin before they were to do an operation. Can somebody confirm? I haven't been in the hospitals. Do they still use quaternary ammonium compounds like before surgery? Nobody knows? Okay.
Is it the orange stuff they put on your skin before they do dentistry?
Iodine. That's tincture of iodine, although it's more of a brown substance.
Yeah,
I'm
like dark
sure,
brown,
I'm
orange.
not sure what quaternary ammonium compounds look like, so I don't know if it stains the skin or not. It probably does because tincture of iodine does, and they used to use that tincture of iodine all the time in hospitals before surgery because it was such a good antiseptic and it didn't tend to harm the patient. However, the quaternary ammonium compounds now are better than tincture of iodine. So there's very few hospitals that still use tincture of iodine, but there could be. I don't know. And then the other thing, for a better term, that can be in the cell under the plasma membrane would be, just draw a blank here, what was talking about. I'm going to have to come back to you on that topic. I don't remember what I was talking about. Probably had something to do with alcohol. Oh, yeah, I know what it was now, and that is tincture of iodine is not as good as quaternary ammonium compounds, so most hospitals have switched to quaternary ammonium compounds bef to treat somebody's skin before surgery. And both tincture of iodine and quaternary ammonium compounds were tolerated very well by the patient's skin. It didn't irritate the patient's skin, and at least most people. And then the good advantage of tincture of iodine is you could see where the patient had been treated to reduce the number of bacteria on the skin. You don't quite reduce it to zero, although with iodine, it's pretty close. It gets it very, very well. Alcohol is not as strong, but quaternary ammonium compounds even stronger. And another way to damage the plasma membrane is by polymyxin B or polymyxin antibiotics. This is an antibiotic, and what it does is it puts a hole in the outer membrane of a gram-negative cell or into the cell membrane of a gram-negative cell. And then in gram-positives, it damages the cell membrane. And that is how this frying pan can kill something like an egg. So it can hear that my grandfather is up. My brother would then dump out the water in the sink and then start fresh again, which I'm not sure why my grandfather would make eggs and test them. This is, you don't need to know. It's just that um, generally with eggs, all you have to do is cook them for 10 minutes. My guess is my grandfather didn't use a timer. It's been too long, so I don't remember. And he would sometimes make us eggs, and sometimes the eggs would be hard boiled, and other times they would be soft boiled. Polymyxin B, the quaternary ammonium compounds and alcohols can all damage the plasma membrane. Polymyxin B puts a hole in it. If the cell is a gram-positive cell, it'll put a hole in the cell membrane. If it's a gram-negative cell, it mainly puts holes in the outer membrane of the cell wall. But the patient can get, or the person can get, bacteria to reside on the hands and stay on the hands, so that even if you're handling something like alcohol, you can still get the bacteria on your hands and do something with it. Any question about any of that? If not, let's move on. The movement of materials across the plasma membrane can only happen in certain ways. And the movement of materials across the plasma membrane is critical for cells. You must have certain molecules entering the cell, or the cell will die. And you must have certain molecules leaving the cell, or the cell will die. A molecule that has to leave the cell, meaning you don't want it in the cell, would be a toxin. And so the cell, if it has the plasma membranes to remove it, you can remove it just by going in and grabbing it and then throwing it in the garbage can, because you obviously wouldn't want that. So that's one way. And that is actually something that happens with carbon dioxide and oxygen. Oxygen tends to diffuse in, and then carbon dioxide tends to diffuse out. The only exceptions would be if somebody was monitoring their blood for certain chemicals, and then they give themselves a certain chemical. If you then freeze the tissue or get the tissue very cold, the instrument may not work. Any question about any of that? All right. There are two processes that cells use for moving materials across the plasma membrane. There are passive process or processes, and this is where the molecule is moving down its concentration gradient. So if the molecule is high outside the cell, it could move passively into the cell and then would stay inside the cell because the molecule is higher outside the cell than it is inside the cell. So that's a passive movement. There are three different types of passive movements. There's simple diffusion, which I kind of described for you already, and that is oxygen is higher outside the cell than inside the cell. So oxygen can move right through the cell membrane, right through the curtain on my window, and it can just do that until obviously the sun sets. And that is simple diffusion, where the molecule just moves into the plasma membrane. There's another type of a passive process, and that's facilitated diffusion. This uses a protein. I'm going to call it a channel protein, but you could call it a transport protein. In one of the slides in this lesson, I think it's even called the transfer protein. And if you have the molecule or the ingredient, like on the frying pan, made up of stainless steel, which stainless steel actually does act as a passive process for moving the molecule into the cell. But facilitated diffusion uses a protein. I'm just going to call it a transport protein, so the molecule can move through the membrane at the channel protein. And then the third type of a passive process happens with osmosis. Osmosis is water. So this third process really only happens to water, where the molecule, the solvent molecules, can move down their concentration gradient and then move into the cell by osmosis. Any question about the two processes or the passive process for moving molecules about in the cell? If not, let's move on to the second process, and that is an active process where for each molecule you move into the cell has to expend energy to make the cell move into the cell, make the molecule move into the cell. But the good news is that this is an active process so that all of the molecules can actually be transported into the membrane. The membrane itself doesn't go in your buckets, at least not when we turn them in for recycling or whatever you do with them. But um, it's an active process, so the molecule moves up its concentration gradient, meaning the molecule moves from areas of low concentration to areas of high concentration. And that's the opposite of a passive process. So it takes work for the cell to move one molecule into the cell, assuming that there's more of that molecule inside the cell than outside the cell, or that the tonicity of the water in the stream will be less than the tonicity of the water at the patient's address. And so if the patient were to spray you with the uh, with water, The water could move into the cell by a passive process, meaning moving down its concentration gradient from areas of high concentration to areas of low concentration. But if, let's say, something like a plant were to be short on water, but that there is more water in the plant than outside the plant could use active transport to transport one molecule of water inside the cell. But this would require two things. First, the cell would have to use ATP to get the energy the cell needs to transport the water molecule from inside the cell to outside the cell. And then the second thing that active transports need is they need a transport protein, which is required to move the molecule from outside the cell to inside the cell. And for each molecule you move, you use up one ATP molecule. Any question about any of that? Hmm. Remember, passive processes are simple diffusion, and that's simply a small molecule, generally not charged molecule, can move into the box and can get trapped in there. My friend who is uh, cooking pumpkin actually discovered that the pumpkin, when he took it out of the refrigerator, not out of the cooking, and wasn't able to work on it immediately, simply wrapped it in plastic, and then the nutrients after the cooking will not dissipate and would be very good sounding. Oh, I want to mention the difference between the simple diffusion, facilitated diffusion, and osmosis. The difference between these three is osmosis only really involves water, and water is a solvent, and the movement of water into cells is required by the cell. But the point is that this movement is a passive process where the water moves from areas of high concentration to areas of low concentration. And then water is a solvent, not a solute. And the other, simple diffusion and facilitated diffusion, are talking about the movement of a solute molecule into or out of the cell. The example I give is into a cell. In a simple diffusion, the cell doesn't spend any energy. It doesn't need a it doesn't need a transport, not a transport. It doesn't need a transport protein. I guess it doesn't need a transport protein. It also doesn't need a membrane to move through. So simple diffusion could simply be where you added water to one side of the soil in a terrarium, and the other side of the soil you didn't add water, but the water will diffuse throughout the soil of the terrarium so that the water will eventually become equal to both sides of the terrarium. And that's a simple diffusion where the solute moves. You do not need a membrane with simple diffusion. You can have a membrane, like when oxygen moves into the cell, it's moving across the cell membrane, but simple diffusion does not require an enzyme. Facilitated diffusion is where a channel protein can help the molecule move into the cell. Facilitated diffusion does require a, a membrane, either the cell membrane or organelle membrane. Facilitated diffusion still is a passive process, meaning the cell does not use ATP to move the molecules into the cell. And that means the molecule can only move into the cell up until the two solutions become isotonic, in which case the pull into the cell will stop and the person could get, I don't know, a headache or something from that facilitated diffusion. Any question about passive process? And then active transport, you always have a membrane, usually the cell membrane, but it could be an organelle membrane. You always have a transport protein, and that's what's moving the molecule into the cell or out of the cell. And then this requires the use of ATP. You use up one ATP for each molecule you move into the cell. I've got an arrow showing on my screen here, and are you guys hearing me okay? Can anyone confirm that?
We
Yeah.
can hear you.
Great. So here's an example of simple diffusion. When you put a drop of, I don't know, ink or something colored, a drop of some colored solution in the water, the drop will eventually diffuse throughout the water. This is simple diffusion, that movement of the dye molecules into the water, shown here, an actual thing, actual transmission electron or whatever, photographic image. So simple diffusion involves a solid moving from areas of high concentration to areas of low concentration. Eventually, all of that dye will totally diffuse throughout the solution, in which case it'll then be a very light shade of purple. It won't be this very dark one there and a semi-dark one there. I just noticed that almost looks like a hand. Let me look that blown up. Maybe. Not important. Just notice that for the first time. A simple diffusion is important for the transport of oxygen into a cell and the transport of carbon dioxide out of a cell. Facilitated diffusion always uses a membrane like the cell membrane. It always uses a channel protein. They're giving the name transporter protein. I don't use that term. I just call it channel protein. And they shouldn't be using glucose because glucose is a poor example. A better example would be fructose. Fructose moves into the cell by facilitated diffusion, coming across a channel protein. But it's a follows the laws of simple diffusion. And once the molecule equals the same concentration outside the cell as inside the cell, then no further molecules can be transported into the cell. There's no room for it because the container containing the raisins is dried. But if you were to put raisins in something like coffee or a liquid, like could be water, fructose can only move into the cell by facilitated diffusion. So once the concentration of the fructose is equal outside the cell than inside the cell, no more fructose can then enter the cell. We're talking about the net relationship, but it's essentially the same as no more fructose can enter the cell. And that's because the fructose has to move down its concentration gradient or else it's not going to move. In fructose, it means if you eat fructose, you would never digest all of them because once the concentration equals the concentration inside the cell compared to outside the cell, then the person could come and grab the container. We have it here so that it could be shut with tape or glue or something like that, and then we recycle it. Glucose doesn't quite work that way. Glucose will initially work that way, where this facilitated diffusion is a channel protein allowing the glucose to move through. However, and it's a non-energy specific reaction. However, once the glucose concentration inside the cell equals the concentration outside the cell, by the laws of diffusion, no more glucose can enter the cell or leave the cell as well, too. And that's because the molecule can only move down its concentration gradient. And if it's no longer higher in one place, it's not going to move to the other place where it's lower. So once the molecule equals its half concentration, then no further molecules can move by facilitated diffusion. Did I talk about why glucose is different? When glucose is at high concentration outside the stomach, glucose will move into the gastrointestinal lining cells by facilitated diffusion. But when the glucose equals outside the cell, equals to the glucose inside the cell, then this channel protein changes and becomes an active transport protein, meaning it becomes an active transport protein. And now the cell doesn't use facilitated diffusion to bring glucose into the cell. It uses active transport, which costs the cell 1 ATP in energy for each molecule the cell moves into the cell. Any question about any of that? You don't need to know about glucose, by the way. I won't quiz you on that. I'm just saying that glucose was not a good example here. All right, we did mention it briefly, and that is with facilitated diffusion, the solute combines with the transporter protein, meaning the facilitated diffusion protein. I'm just going to call it a core protein or a channel protein, one that has a pore in it, allowing the solute to move into the cell as long as the concentration of the solute is higher outside the cell than inside the cell. And I do need to say that it's not any molecule. Generally, it has to be that one molecule has to be inside the cell before you can open the cabinet door with just your keys. Obviously, the firemen are really good at opening doors. So the passive process happens in both simple diffusion and facilitated diffusion. In osmosis, though, this is not a solute, it's water, a solvent. Water can move selectively across the plasma membrane from areas of high concentration to areas of low concentration. But with green plants, they can also use a transport protein to move water into the cell, in which case water can then move into the cell much quicker. All right, any question about any of that? Already talked about that. Solute concentration may move down its concentration gradient as long as there's a transporter protein, or I'm going to call it a channel protein, to allow the molecule to move into the cell. Oh, come on. Bacterial cell can experience one of three different kinds of osmotic solutions. There's an isotonic solution, a hypotonic solution, and a hypertonic solution. Note that these are relative terms, so it's best to say something like the solution outside the cell was hypertonic relative to the solution inside the cell. If you're only going to say something like the solution outside the cell was hypertonic, that's not quite good enough. Any question about any of that? All right, the easiest to explain is an isotonic solution, and that would be where the molecules outside the cell are equal to the molecules inside the cell. So there's no net movement of these waters into the cell and no net movement of this molecule out of the cell. Why that's happening is because the molecule is higher at concentration outside the cell than inside the cell, in which case the molecule can move into the cell. But if in an isotonic solution, we don't have that, and then there's no net movement of water either into or out of the cell. The cell is generally happy because it's an isotonic solution, so it's not going to die of osmotic burst. Any question about any of that? I guess I need to mention that for osmosis, it can be dangerous to the cell if the cell wall does not have DNA already incorporated into it. Any questions about this? The cell can also find itself in a hypotonic solution. This solution has lower solute concentration outside the cell than inside the cell. And so there will be a net movement of water from outside the cell to inside the cell. Note that the concentration of water is, I don't know, 180 degrees different than the concentration of the solute. So here we have a low solute concentration outside the cell, a high solute concentration inside the cell, so that the solute would want to leave the cell where the concentration is higher. Hypotonic solutions can be dangerous for the cell if the cell does not have a cell wall. If it's not present, then the cell gets on an antibiotic which causes danger or damage to the cell membrane. The damage will be done, and the cell generally cannot repair it, and then eventually the cell will die. It may take 6 months or longer to die, but eventually it will die. So in a hypotomic solution, there's net movement of water into the cell. So there's more water outside the cell, less water inside the cell. Sorry. Um, sorry, I'm grading now. I didn't mean to answer this. I'll try and call you back later. Um, where was I? Hypotomic solution is higher water outside the cell than inside the cell. So there will be a net movement of water inside the cell. Where there's water, high water, that means there will be low solute concentration. So there will be low solute concentration outside the cell because there's more water outside the cell. And then there will be more solute inside the cell where there's less water. If the cell is in a hypotonic solution, then water will move into the cell, which will swell the cell membrane. And if there's no cell wall, the cell membrane will continue to expand until the water moves into the cell enough that it'll actually burst the cell membrane, causing the cell to die. If the cell has a functional cell wall, the cell membrane can only expand up to the cell wall, in which case the cell membrane cannot expand any further. That will limit the water from moving into the cell because there's no room for it. So no more water will move into the cell. And that's how the cell wall can protect the cell and prevent osmotic lysis. However, if the cell wall is already damaged, like been exposed to penicillin G, where there's a hole in the cell wall, or the cell wall has been damaged by the enzyme lysozyme, then the cell membrane can come out the hole and continue to expand until it bursts. Most bacteria live in a hypotonic solution. At least I should say most bacteria that infect a person, most pathogenic bacteria. So if a bacteria gets inside of you, you're have more water than the bacteria. And that's why penicillin G and the other members of the penicillin family can actually work, killing the cell. So I guess one way to say it is that it's not penicillin G that directly kills the cell. It allows the water to come into the cell to kill the cell. Any question about any of that? All right. The third solution that a water, or I should say environment that the cell could find itself is a hypertonic solution. This has higher solute concentration outside the cell than inside the cell. So there will be a net movement of water outside of the cell that will cause the shrinkage of the cytoplasm of the cell, where the cell membrane will actually pull away from the cell wall as water leaves the cell. You can actually view that. We used to see that in biology 161 or 160 in your preliminary class, where you put a cell into a high hypertonic solution, high solute concentration, so the water would then leave the cell, and you could watch the cell membrane start shrinking, pulling away from the cell wall. Normally, the cell membrane is up against the cell wall, so you can't see them apart from each other, even using a light microscope. But once it starts shrinking away from the cell wall, you can then see the cell membrane. And that can lead to plasmolysis, where so much water would leave the cell that that would kill the cell. It depends on how sensitive the cell is to plasmolysis for how long the cell can survive. But something like E. coli is fairly sensitive to the cell, and I don't know how long it takes for the E. coli to die, but it would be within only a couple of days, or even less than that, maybe a couple of hours. If the cell has a lot of mycolic acid in the cell wall, that will prevent the water from leaving, and then that cell would survive much longer in a hypertonic solution. And that is why the Mycobacterium can survive much longer in a hypertonic solution than most other bacteria. Obviously, an exception would be an endospore, and it can survive much, much longer than the Mycobacterium, because an endospore could survive, like in the deserts of Egypt. We've had them surviving, and then they germinated them for over 8,000 years. The cell was in the endospore state. It was still viable after 8,000 years. I think the longest endospore we've gotten to germinate was one that was from a drilling in the South Antarctica, and they took out the ice at different layers, and that's how old the ice is. The deeper down you go, the older it is. And they got some endospores to germinate after they had been frozen for something like 40 million years. No, not 40 million, 40,000 years, 40,000 years, sorry, not 40 million, 40,000 years. So a hypertonic solution generally leads to cell death. And that's why when you open your bottle of molasses, when the bacteria and other microbes fall into the molasses from the air, it's not a problem. And you can keep your molasses for years at room temperature. And it's because molasses is a hypertonic solution. If you want to know how long you can keep your molasses, it's at least 30 years. My mother had a bottle of molasses that we figured was at least 30 years old. It hadn't been used for at least 20 years. No, the molasses was used for 20 years. I was the last person to use it. I made some molasses candy for my father, and he ate it and he loved it. So obviously the molasses was good that we know of for at least 20 years, and it was probably good for 30 years. But my mother said, I'm not using this molasses. You're not using it. It's over 30 years old. I'm going to toss it. So we finally got rid of the molasses. Any question about any of that? All right. Active transport is great when the concentration of the molecule that's needed is higher. Is it high enough concentration outside the cell than inside the cell? And it's also great when the concentration of the molecule is higher outside the cell than inside the cell, because the molecule can then move into the cell by a passive process. But what do you do with a cell in the deep sea or a cell in the Sahara Desert? The concentration of the molecules the cell is going to need will be much less outside the cell than inside the cell. Like the concentration of glucose in the water is going to be very dilute, and there will be much more glucose inside the cell. The same thing with the cell inside the Sahara Desert. So what does the cell use? It uses active transport to move the molecule into the cell, even though the molecule is at lower concentration outside the cell and at higher concentration inside the cell. This requires the cell to spend energy to move the molecule into the cell because it takes the cell work to move the molecule from an area of low concentration to an area of high concentration. Cells use active transport to pump the solute molecule against its concentration gradient. So we have, first of all, we have a membrane, like the cell membrane. We have a transport protein inside the cell membrane. And then that transport protein can bind to generally one specific molecule, and allows only that one specific molecule to enter the cell. And to get that molecule to move into the cell, you must spend ATP, because we need energy to move each molecule into the cell, because we're moving the molecule against its concentration gradient. Generally speaking, these transport proteins are molecule specific, but if you know anything about drugs, we do have some drugs that mimic the substrate for the transport protein. And so you can give the patient the drug, and then the drug will use up all the transport protein so that the molecule is not transported into the cell. Any question about any of that? So, active transport is found in all cells, eukaryotes included. Group translocation is a special form of active transport that is only found in prokaryotes. Group translocation is similar to active transport. It does have a transport protein which pumps the molecule against its concentration gradient. You do need energy to move the molecule into the cell. However, with group translocation, we usually don't get that energy from ATP. It's obtained from a high energy phosphate compound. And actually, ATP is a high energy phosphate compound. But my point is that group translocation tends to use another high energy phosphate compound, such as PEP. And then glucose is transported into the cell with group translocation. And when that glucose is moved or transported, the glucose is chemically altered. The phosphate group from PEP will be transferred from PEP to the glucose. And this alteration prevents the escape of the cell, the molecule from the cell, as well as making it easier to pump the molecule into the cell. Let me quickly show you with paint. Come on. So remember, we're talking about group translocation. So here's the cell, here's the molecule outside the cell, and there's more molecules inside the cell. So let's make them 10 molecules inside the cell. I should have done something easier. That's 9. And now we have 10. And let's say we have 10 outside the cell. Let me do something that's a little easier. Which I guess I shouldn't because it's the same molecule, so we'll leave it there. And we have 10 outside the cell. I'm just going to write that to make it a little easier. All right. If we use only group translocation to move these molecules into the cell, then the last molecule to be moved in, there will only be one outside, and then there will be 19 inside. You're going to be moving one, which is 1 out of 1, to adding 1 to make it 1 out of 20. And that's a very steep gradient, meaning there's much fewer outside the cells than inside the cell. And so that will need to take a lot of energy to move that molecule and transport it into the cell. Any question about that part? If you use group translocation, you don't have to move all 10 of them by group translocation. You can move, oh, I still got it there. You can use 5 translocation and move 5 by active transport. Now, instead of being 1 to 19, well, let's make another one. So instead of being we now are moving 1 well, it's kind of 1 out of 5. And then the active transport is moving the other 5. And so this 1 will be moving 1 out of 1 to 1 out of 5. That's a much less steep gradient to move it against than you did if you moved all of them by active transport. Any question about any of that? Anyways, that's trying to show you why prokaryotes will move molecules both by active transport and group translocation. And the difference is that active transport will not change the molecule. If this is glucose, the glucose will be moved into the cell as glucose. But if it's active group translocation that's moving the glucose will be changed and it will be moved into the cell as glucose 1,6-phosphate. So it's not glucose being moved in the cell, it's glucose 1,6-phosphate. Any question about any of that? All right, any questions at all about this lesson? If not, I'm going to stop here. I think we're at the ending time, and I will see you in the next lesson. All right, goodbye.